Okay, hi guys. This is the review for the midterm part 1. I'm going to start with the cellular energetics part because the exam is going to be heavily focused on that in lieu of having another unit exam right before your midterm. So we're going to start with that, and then we'll go back to some of the other topics after that. So similar to the other videos, I'm going to go through some questions with you so you can practice thinking those through, and then at various points I'm going to stop and kind of return to my Word document here just to type out quickly for you things that I hope help you highlight how to study. Okay, so let me switch screens here. All right. So this is from that same review site on Biology Junction that I've shared with you before. Here, number 2 is a basic definition question, but I think you'll see in a second why I decided to go over this one. Which term most precisely describes the cellular process of breaking down large molecules into small ones? Catalysis, metabolism, anabolism, dehydration, or catabolism? So I thought this was kind of a tricky question because they're asking something really specifically where, you might have thought about arguing that maybe that metabolism was it. And then if you ruled that out, you're going to be kind of stuck between catalysis and catabolism. So let's look at metabolism first. I'm just going to show you on Wikipedia here. All right, metabolism is the set of chemical reactions that happens in the cell of living organisms to sustain life, blah, blah, blah. Metabolism is divided into 2 categories. Catabolism breaks down organic matter, okay, and anabolism uses energy to construct compounds. So metabolism includes both catabolism and anabolism, okay? Now, catalysis, okay, is a reaction that uses catalysts, enzymes, okay, and has to do with the change in rate. So that one's definitely out. So let's look at the question again. Which term most precisely describes the cellular process of breaking down large molecules into smaller ones? Well, the answer is going to be E then, okay? So the metabolism includes both anabolism and catabolism, okay? And catalysis is different, okay? It has to do with enzymatic reactions. All right, number 21. ATP generally energizes a cellular process by releasing heat upon hydrolysis, acting as a catalyst, coupling free energy released by ATP hydrolysis to free energy needed by other reactions, breaking a high energy bond, or binding directly to the substrate of the enzyme. Oops, I gave away the answer there, moved a little bit. Okay, so the answer is C, coupling free energy released by ATP. Okay, energy coupling is a concept that you should know. If you forget what energy coupling is all about, let me see if I can show you. Okay, here's the definition for energy coupling. Look at number 2. Free energy from ATP hydrolysis is coupled or functionally linked to the energy needs of another chemical reaction. That's part of how ATP works, through energy coupling. Okay. Okay, number 22. What term is used to describe the transfer of free energy from catabolic pathways to anabolic pathways? Okay, so from breaking down to more of a synthesis. Okay, feedback regulation, bioenergetics, energy coupling, entropy, or cooperativity. What do you think? Ding, ding, ding. Energy coupling. Okay, so that's what we were just talking about. All right, so here's the big concepts we're hitting so far. ATP, know the structure and function. Energy coupling, catabolism, anabolism, metabolism, and then endergonic and exergonic, knowing the difference. So just to kind of keep you up with what things you need to know so far, those are them. Going back to the questions now. Number 24, which of the following statements regarding ATP is or are correct? ATP serves as a main energy shuttle inside cells. That sounds kind of good. ATP drives endergonic reactions in the cell by the enzymatic transfer of the phosphate group to specific reactants. Well, for that one, I've got to know a bunch of this vocab, and I definitely have to know what endergonic means. I'm going to have to return to that one. The regeneration of ATP from ADP and phosphate is an endergonic reaction. There's endergonic again. Let's see if I can find out what endergonic means first. Okay, so the answer here, I'm sorry I had to pause for quite a while there. I'm making this during that snowy day, and dogs need to come out, get in, be dried, etc. Anyway, the answer to this one is E, okay? A, B, and C. So it's all of these things. So this is probably a good one to kind of pause and make some notes on that, that these are all a part of what you need to know about ATP. All right. Number 26. By the way, we're still all in that chapter on ATP, chapter 8, I think. Okay, how can one increase the rate of a chemical reaction? A, increase the activation energy needed. B, cool the reactants. C, decrease the concentration of the reactants. D, add a catalyst. E, increase the entropy of the reactants. So there's a bunch of vocab words being tossed around. So the answer is D, add a catalyst. So what's a catalyst? All right, let's go over here. All right, enzymes. Sorry, that was my phone. Enzymes are organic catalysts consisting of proteins. Okay, so enzymes themselves are catalysts. Catalysts are molecules that speed up reactions by lowering the energy of activation, meaning it makes it easier to get a reaction started. Okay, activation energy is the energy required to activate a reaction. So enzymes lower the activation energy. Okay, catalysts are not used up in the reaction but can be reused again and again. By using enzymes, cells control when, where, and how fast the chemical reactions that are useful to the cell will proceed. Enzymes are critically important for living things. And when we talk about anatomy and digestion, we'll talk about the role of enzymes in digestion as another huge part of what enzymes do. Okay, make it easier to break down food into energy. All right, so the substrate, just to throw in all the vocabulary at once here, right, is the reactants acted upon by an enzyme are called substrates. Okay, so you have an enzyme and a substrate. The enzyme is acting on the substrate. And here this little image is supposed to show you that, but I think it's all messed up, but I just liked how this author had bulleted these. This is from Mansfield, Ohio State .edu, and the professor's name is Sabadon, but it's just an outline of the Campbell biology book. Enzyme specificity, we talked about in class that enzymes know exactly what they're supposed to do, and they're very specific for the reactions that they do. So let's go back to the question then. Oh, we answered the question already, that it was D to add a catalyst, and then we went over catalysts. So, ding. Okay, enzymes, what do you have to know? What are they? What is activation energy? What's a catalyst mean? Substrate, reactants, and variables that affect the enzymes. So, temperature, etc. So you might want to pause right there and get down the big things you have to know about enzymes. All right. do expect you to know about the active site, the induced fit, and the complex that's formed. Okay? I'll write that down for you. Oopsie. It's awkward because you can't move the video window around once you have it set up. I can't move this up further than that. There we go. Okay. So this should say, okay, so those are big concepts there for enzymes. Back to our questions. Reactants capable of interacting to form products in a chemical reaction must first overcome a thermodynamic barrier known as the reactions. That's the part you can't see. After as, it says the reactions. Entropy, activation energy, endothermic level, heat content, free energy content. If you said activation energy, you'd be correct. Okay, so that's pretty much the definition of activation energy, is the energy required to overcome the thermodynamic barrier. Okay, number 34. Many different things can alter enzyme activity. Which of the following underlie all types of enzyme regulation? A. Changes in the activation energy of the reaction. B. Changes in the active site of the enzyme. C. Changes in the free energy of the reaction. And then we have an A and B and an ABC. So what do you think? Right, so the answer is D, A and B only. So we can cross out C because the free energy of the reaction is not involved here. It's not changed. Okay, so what does an enzyme affect? It affects the activation energy, and it does cause a change in the active site of the enzyme. Okay, number 38. Which of the following is true of enzymes? Enzymes may require a non-protein cofactor or ion for catalysis to take place. B. Enzyme function is reduced if the three-dimensional structure or conformation of an enzyme is altered. C. Enzyme function is influenced by physical and chemical environmental factors such as pH and temperature. D. Enzymes increase the rate of chemical reactions by lowering activation energy barriers. And I chose this question because I thought it kind of gives you a big picture about enzymes. It gives you a lot of stuff in a short question. Okay, so the answer is E, all of the above. So this question is a great summary of things that you have to know about enzymes. Okay, so first of all, you can have a non-protein cofactor, and what's that called? It's called a coenzyme. Enzyme function is reduced if the structure is changed. Well, remember that for proteins, structure is everything. Okay, so the structure is super important. If you don't got the structure, you don't got the function. Enzyme function is influenced by pH and temperature. Well, definitely, and I believe you saw that exactly in your lab. Enzymes increase the rate of chemical reaction by lowering activation energy. Well, that's the definition of an enzyme. So this is the really great question that chunks everything you have to know about enzymes. So I would definitely jot this one down. 53. What is the term used for the metabolic pathway in which glucose, C6H12O6, is degraded to carbon dioxide and water? So cellular respiration, glycolysis, fermentation, the citric acid cycle, or oxidative phosphorylation. Well, right off the bat, I want to say that if you are not familiar with the definition of any of these 5 terms, write that down right now of which one you need to review, because all 5 of those terms you are responsible for. All right, so the answer here, of course, is A, cellular respiration. All right, so let's start making a list of the things that you have to know about cellular respiration. All right, so you need to know the formula in words and symbols. Right, so that would, if you know that, that helps you answer that question we just looked at. Be able to explain how each reactant is used and each product is formed. So be able to fully grasp all of what's going on in that formula. Okay. Number 55. Which of the following statements is or are true, correct about an oxidation reduction or redox reaction? The molecule that is reduced gains electrons. The molecule that is oxidized loses electrons. The molecule that is reduced loses electrons. The molecule that is oxidized gains electrons. E, both A and B are correct. So this, if you have some trick already, hopefully you do, like the oil rig or whatever, can quickly figure out what the answer is. And it is E. A and B are correct. So the molecule that is reduced gains electrons. The molecule that is oxidized loses electrons. Okay, so now applying that redox reaction concept, which of the following statements describes the results of this reaction? In this box here, that's where the arrow should be. For some reason, some of the graphics just don't appear right on this review site. So hopefully you recognize this formula that we're looking at, which is cellular respiration. They want to know what's being oxidized and what's being reduced. So instead of reading all that, I'll give you a minute to take in the magic. Okay, so the answer is A, okay, that the glucose is oxidized and the oxygen is reduced. All right, so it's just being able to apply the redox reactions to cellular respiration. Okay, so number 61. Which of the following statements about NAD plus is false? And I want you to know that while I was just looking through to see what question I wanted to look at next, that I was really stuck looking at this question and I couldn't figure out why the answer that they had was the right answer, and I was just going to skip it, and then I realized it was me not reading false. So I thought they were asking which was true, and they're asking which is false. So this is something that can really trip you up in multiple choice exams, and it's something that has always been hard for me. I hate questions like this, but unfortunately, in order to prepare you for the AP exam, we give you questions like this. All of the following are true except, or whatever. So I would always give something like false a big circle or put a star by it or something just so it doesn't confuse you the way it just caught me, because I got really confused. So I'll let you read this, pause it, and then I'll show you the answer. All right, so the answer is B. Okay, NAD plus has more chemical energy than NADH. And so given that I thought that they were asking you which is true, I was very confused because of course, okay, NADH has a lot more energy than NAD plus, because the whole point of this molecule is to carry high energy electrons around to needed places for reactions. So once it's into NADH, it's like it's one of those piggy banks, all right? It's a hot ticket. It's got a lot of energy, a lot more energy than NAD plus. Okay, where does glycolysis take place? The matrix, the outer membrane, the inner membrane, the intermembrane space, or the cytosol? Well, of course you know this one. This one's an easy one. The cytosol. Okay, because even prokaryotes can do glycolysis, and they don't have mitochondria. So for this part of cellular respiration, about what you have to know, we already talked about having to know the formula and words and symbols and being able to explain how each reactant is used and each product is formed. You'll have to know where glycolysis takes place, where the citric acid cycle takes place, where is the electron transport chain, and be able to draw and label a mitochondrion. All right, there's more that you have to know, but that's the first part. Number 69. In addition to ATP, what are the end products of glycolysis? Carbon dioxide and water, carbon dioxide and pyruvate, NADH and pyruvate, carbon dioxide and NADH, water, FADH2, and citrate. What do you think? Pause at this point. Okay, now I'm going to give you the answer. The answer is, of course, C. Okay, which brings us to the other stuff you have to know. Okay, oops. The products of each part of cellular respiration. Right, so that's super important. Okay, number 74. How many carbon atoms are fed into the citric acid cycle as a result of the oxidation of one molecule of pyruvate? All right, so this could trip you up a little bit, okay, because you know that after glycolysis, glucose is divided into 2 molecules of pyruvate, and that each molecule of pyruvate has 3 carbons in it. But what's the transition from pyruvate into the citric acid cycle? So how many carbons are going to move forward? Well, that's about that intermediary step. And let me take you to a little picture here. Right, this, hopefully you can see it okay. I thought it was a pretty good summarizing picture of everything all in one picture. But so here's our 2 molecules of the 3-carbon pyruvate going from the cytosol into the mitochondria, and in that intermediary step, remember, carbon dioxide is given off. So that acetyl-CoA is a 2-carbon molecule that goes into the Krebs cycle. Okay, so the answer then is 2. Okay, so when pyruvate is transported into the mitochondria, carbon dioxide is given off, and acetyl-CoA is what goes on to the Krebs cycle. Number 78. How many molecules of carbon dioxide would be produced by 5 turns of the citric acid cycle? Well, this is pretty easy as long as you know what's produced by 1 turn of the citric acid cycle. So if you are forgetting that, I'll show you a little review of that right now, and we'll return to the question. Oh, that's not it. There we go. Okay, so here's a summary of what's produced by the Krebs cycle. So you might want to pause and write this down. 2 carbon dioxides, 3 NADH plus proton, or hydrogen ion rather, 1 FADH2, and 1 ATP. So you might want to pause and jot that down. Okay, so if we know that 1 turn of the Krebs cycle yields 2 carbon dioxide, 5 turns will of course give us 10. Okay, and don't forget as you're answering questions that whether they're asking you per molecule of glucose or per molecule of pyruvate, but keep in mind that, you know, for 1 molecule of glucose, you're going to go through the Krebs cycle twice. Like, just always keep that in the back of your mind. So now that you're reminded about that, let's do another one like that. Starting with citrate, How many of the following would be produced with 3 turns of the citric acid cycle? So they're not asking per molecule of glucose, but they could. This time we're just asking about the cycle. So pause for a second and then think about it if it's going to be 3 turns. And the answer is D, 3 ATP, 6 CO2, 9 NADH, and 3 FADH2. So make sure that you know the products of every part of cellular respiration. Number 81, carbon dioxide is released during which of the following stages of cellular respiration? A, glycolysis and the oxidation of pyruvate to acetyl-CoA. B, oxidation of pyruvate to acetyl-CoA and the citric acid cycle. C, the citric acid cycle and oxidative phosphorylation. D, oxidative phosphorylation and fermentation. E, fermentation and glycolysis. So here's a pretty critical question. If you remember, okay, one of the things I said you should be able to know about cellular respiration is be able to explain how each reactant is used and each product is formed. And if you can do that, that really gives you the whole picture of cellular respiration. And this question is really asking that, for you to know exactly where carbon dioxide is released. So the answer is B, oxidation of pyruvate to acetyl-CoA, which is the intermediary step between glycolysis and the Krebs cycle, okay, and the Krebs cycle, the citric acid cycle. You definitely need to know both terms, by the way, because a lot of books still use the Krebs cycle. All right, and this I thought was a really good question. Along the same lines of, as I said, to emphasize knowing the parts of cellular respiration, the parts of the formula, and being able to explain how things are used will give you the whole package. During oxidative phosphorylation, water is formed. Where does the oxygen for the synthesis of the water come from? Okay, and here are our choices. All right, well, if you think about the formula in general, okay, these two, B and C, are on the left side of our equation, and they both have significant amount of oxygen. So which oxygen is going to be used when that water is formed? Okay. And the answer is C. It's the molecular oxygen, O2. All right, so the water that's formed is using the oxygen that we've breathed, okay? And the CO2 that we breathe out, the oxygen in the CO2 is the oxygen coming from the glucose when the glucose is broken down. So that's how that plays out. Okay, number 90. Energy released by the electron transport chain is used to pump hydrogen ions or protons into which location? Okay, so you can, they're not trying to mix together the photosynthesis and the cellular respiration for this question. It's really just about the cellular respiration. And so they want you to be able to know, like we're getting the buildup of protons somewhere and then they're being pumped somewhere, and where is that happening? So the answer is D, the mitochondrial intermembrane space. So let me just remind you of what you have to know about ATP synthase. Okay, for cellular respiration, the protons are going from where to where, and for photosynthesis, the protons are going from where to where. You need to be able to answer that question. Okay, because remember, it's kind of different out and in for each. So spend some time going over that. All right, so in this case, for cellular respiration, they're being pumped into the intermembrane space from the matrix, where Keanu Reeves lives. Okay, number 91. During aerobic cellular respiration, a proton gradient in mitochondria is generated by blank and used primarily for blank. I think this is an awesome question to pull these ideas together. So how is the proton gradient being formed and what is it being used for? And I would say, to make sure that you know the answer to this question if we were talking about photosynthesis too. So the options are the electron transport chain and ATP synthesis, electron transport chain and substrate level phosphorylation, glycolysis, production of water, fermentation, and NAD plus reduction, and diffusion of protons and ATP synthesis. So pause a second and then I'll give you the answer. Okay, the answer is A, the electron transport chain and ATP synthesis. Okay, so pretty cool. That's a great question for pulling these ideas together, and of course you should know what the rest of these are all about too. Okay, number 105. In the absence of oxygen, yeast cells can obtain energy by fermentation, resulting in the production of, and I'm not going to read all those in the interest of time. Of course, this is about fermentation, and we talked about the two types of fermentation, alcoholic fermentation and lactic acid fermentation, so they want you to know which one yeast does. And I'm going to show you the answer in a second, so you might want to pause and think about it if you don't know yet. And the answer is A, okay, ATP, CO2, and ethanol, because it's alcoholic fermentation. Right? You do need to know both types of fermentation. Alright, so in the interest of making these videos not so crazy long, I'm going to end this one before I go over the parts of photosynthesis. But just to remind you about, what we talked about here of some key points you need to know about cellular respiration. Also here, for each of the parts of those glycolysis, the Krebs cycle, and the electron transport chain, you need to know what's going in and what's coming out. For example, knowing that with glycolysis we're talking about glucose in and 2 pyruvate out, and then also how many ATPs. So be able to keep score of the ATPs. Okay. So those are the big main points. There's something else I was going to say and I forget. Alright, well, I'll go into it in the next one because we'll continue from there. I hope this was helpful. It's always good to practice questions and take notes on what you don't know, review, and then take a look at these questions again. You can just kind of skim through the video and look at the questions, or go back and look at the questions on Biology Junction website, biologyjunction.com, and go to the AP Bio section, and then you'll see I think they're called test guides or study guides. Okay? All right, that's the end of part 1 of our review.